No? Okay, so I'm gonna kind of give you an overview of chapter 7 and I'll continue maybe with chapter 8 after. So I want to start with this slide here. I will be asking you questions. I'm not going to lecture, it's more of a discussion, so try to participate. Otherwise, I'm gonna start calling out names. So can you guys tell me difference between chromosome and chromatin? You guys remember chromatin? So what is, if you had to define chromosome versus chromatin, how would you define that?
Chromosome would be like a portion of the chromosome, right?
Yes, so chromatin, it has all the associated proteins, and so the way your book defines it, chromosome is the DNA and the proteins in eukaryotic organisms. And then the way your book defines chromatin, it says DNA plus the proteins in the nucleus, so inside the nucleus. So I don't know, the only difference I guess is inside the nucleus. So that's how it's defined. Do you guys remember what this was called? What is this or and this here?
Sister chromatids.
Sister chromatids. Can you guys define sister chromatids?
It's a pair of chromatids that contain the same sequences of DNA for similar genetic traits.
Yes, and they are attached right here in the middle at the centromere. That's something that's also common. So they have the same genetic information, they're identical, and then they have the same genes or the gene sequences, like Daniel said, and then they are attached here at the centromere. So this picture here is when you start zooming in. So this would be chromatin. So you have chromosome, looks like letter X, and then you have the proteins here, and you zoom in more, you have the DNA, you zoom in more, you can see the gene. Let me show you a picture. So here you have your chromosome, you start unwinding it, called chromatin, and then you start seeing the proteins here. You zoom in a little bit more, then you zoom in a little bit more, you can see the double helix. And if you zoom in more, you are able to see the nucleotide. So that's the way it goes. It goes from the chromosome to the base pair. So you're zooming in more and more and more, and you're able to see more stuff there. Yes, two said chromosomes contain two chromatids. So can you guys tell me the DNA structure? What is DNA composed of? What is this double helix composed of? Nucleotides, that's one of them. What else? So nucleotides are A's and T's and G's and C's. What else? Devin, what is DNA composed of?
That's what I have down, just nucleotides with A, T, C, and G, like adenine, thymine, guanine, cytosine.
How are they attached?
Is it ligase? Is it that?
No.
Oh.
Anybody else? Daniel?
Or is it
Nancy?
peptide bonds?
Hydrogen
No.
bonds.
What did you say, Daniel?
Hydrogen bonds.
Hydrogen bonds. They're held together with hydrogen bonds. Are they weak bonds or strong bonds?
Weak bonds.
Weak bonds, because they need to be open in order for you to read the information. You guys just know this part. You don't know this part yet. So it's phosphate group and a sugar. That's the backbone. And then you have the bases here. So if you look here, this backbone is phosphate, and then it has sugar. And that gives it the double helix. And then you have your nucleotides, and they're held together through these hydrogen bonds. These hydrogen bonds are weak bonds, and they are covalent bonds. So if I ask you what type of bond is this, then you're gonna say covalent bonds for this DNA. Okay, can anybody define the central dogma of biology? There is a question on the exam. Central dogma of biology. It's on the slide that you can see right now. Now ask Oscar or Kaylin. Kaylin, were you about to say something?
That it explains transcription translation for
Yeah,
DNA.
so central dogma is going from this DNA to RNA to protein. It's not only DNA to RNA or RNA to protein, it's the way genetic information flows, and the genetic information flows from DNA to RNA to protein. That's the central dogma of biology. Make sure you know this. Can you guys, if I were to ask you to define transcription and translation, can you do that? So define transcription for me and the location of it.
Transcription produces an RNA molecule that is complementary to the DNA.
Yes, and translation?
Is the assembly of the amino acids in the cytoplasm.
Yeah, so going from RNA to protein, RNA carries the information to make the protein, and that's done right here. What is the location for both of these?
Transcription happens in the nucleus and translation happens in the cytoplasm.
Translation in the cytoplasm, and if you had to be very specific, you would say happens inside this rRNA molecule. That rRNA molecule is an enzyme because it's doing work. We will get to mRNA, tRNA, and rRNA as well. Okay. What are some differences between DNA and RNA?
DNA is ribose OC and RNA is ribose. DNA is double helix, RNA is single template strand, and DNA is connected to thymine, the other one connected to uracil.
Yes. So this is very, very important because you will be doing some work tomorrow, or not tomorrow, Thursday, when you do the exam, where you have to convert some of the sequences that I give you. So double-stranded means a lot more stable. This is simple, but it's not that stable. So if I give you a question, I give you a sequence, and I say, this is a DNA sequence. What is the complementary DNA strand? You're going to use this. You're going to use T for A, or A for T, because you're going from DNA and you're creating a complementary strand for DNA. If I were to say, this is the DNA strand, give me the RNA strand, then you're going to use uracil for adenine because RNA does not have thymine. So make sure you guys can do this, and it's probably going to be worth more points than the other questions. And then you have three types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Can somebody tell me the functions of these?
Messenger gets information from the nucleus, then process it in the ribosomal, then transfer RNA will transfer it in order to start making protein.
Yes, so messenger RNA, as it's in the name, it takes the message from DNA, convert into RNA, and brings it outside, brings the message outside. You have the information to make the protein. This is where the protein is being made, in the rRNA, so it's an enzyme. And then you have the tRNA. tRNA is going to transfer the correct amino acid to the codon. Can anybody define codons? How many letters are codons?
3 pair of mRNA.
So three letters, 3 nucleotides make one codon. I can give you a sequence where I say count how many codons are there. So you have to know 3 letters make one codon. Or I can say count the codons between the start codon and the stop codon. I will give you the chart, but you have to kind of look which one is stop and which one is start, and you count in between. So make sure you guys can do that. One more thing, actually, I think it's in the next slide, never mind. There is an enzyme I asked you guys to remember for transcription. Anybody remembers which enzyme is needed for transcription?
Ribosomal RNA.
So RNA polymerase. RNA polymerase is the one that's going to add the complementary base pairs. So that's important and that's needed for transcription. Let me show you, actually, before I do that, I want to compare this. What is the difference between mature mRNA and this mRNA?
The mature mRNA is finished, right?
Yeah. So
It doesn't have
what
the
is
cap and the tail.
cap in the tail? So it has the protection. What else?
The introns are removed.
The introns are removed. So this is called mature mRNA. It's ready to go out. Introns are removed and these are added. If you compare this mature mRNA with this DNA, what you can see in the lengthwise, what is different between DNA and this mature mRNA? Yes. So Kawa said DNA strand is longer. Again, that's because it still has the introns. So I can ask you to compare this with this or this with this or otherwise compare this with this. So make sure you kind of know the differences between these. And then here is showing you the codons. And I said 3 letters make 1 codon. You have 1 codon here, another one here, another one here. So you have 3 codons on this DNA template strand. Each codon stands for amino acid. How that amino acid gets there is through tRNA. The tRNA is going to carry the correct amino acid and anticodon, and it's going to bring it to the mRNA sequence inside the rRNA molecule. So here is your mRNA, and this is your ribosomal RNA. It's happening inside this. And then this is your tRNA bringing the amino acid with the anticodon. You find the codons on the mRNA molecule, mature mRNA molecule. You find anticodons on the tRNA molecule. So it was on your quiz too. There was a question about this. And some of you picked that tRNA brings the codon. It doesn't bring the codon, brings the anticodon. Codon is located on mRNA. And this blue part is showing you that it's happening inside of ribosomal RNA, rRNA. It's gonna start at the start codon, it's gonna stop at the stop codon. And in your body, not just one ribosome is working, there's multiple. This is happening simultaneously. And it takes lots of energy to make these. So your body is not making this all the time, only when it's needed. Can you guys define a mutation?
Mutation is a change.
It's a change in a nucleotide sequence. Can you guys define a silent mutation? What is a silent mutation? Devin? I think you're talking, but I can't hear you. You're still mute.
I'm just looking through my notes right now.
Okay. So tell me what silent mutation is.
That's when a change occurs, but it doesn't change
It doesn't change what, Oscar? I couldn't hear the last part.
the phenotype.
The amino acid. So here, this is what Oscar is saying. He is saying the nucleotide is changing. So let's say you have UCU. This is your original sequence. And the nucleotide changed to C. This one changed to C. Now you have UCC. It still stands for the same amino acid. So he didn't mean, I think, phenotype, he meant amino acid. So it's still the same amino acid, even though this changed. So it's a silent mutation. It didn't have impact on the product. You still have the same serine amino acid, but this letter changed. If this letter changed to maybe something else, maybe here, if you had CAC and then C changed to A, now you have a different amino acid. This is not a silent mutation. It's going to have an effect. So this is a silent mutation. What about a frame shift mutation? Can you guys define that?
Is
Insert,
that
Frame
where
shift is sorry. Go on.
I think it's insert one amino acid. It's gonna
So you
cause.
insert one, but it changes the whole sequence. And now you don't have any amino acid that's the same after this got inserted. So it's the most dangerous type, and it's insertion that changes all of the amino acids. So I can give you original sequence, which is not going to be an alphabet. It's going to be DNA sequence. And then I can make a change. I can do maybe change a letter. I can insert something. I can delete something. So you have to kind of compare the sequences. Which type of mutation is this? Or I can ask you the definition of a mutation. And when I say definition, I mean different types of mutations: frame shift, insertion, deletion, silent mutation. Another thing I can do on the exam, I think I forgot to tell you guys, is 64 questions, multiple choice, 63 multiple choice questions, and 10 true and false questions, and you're gonna have 90 minutes for it. Try to log in a little bit early so you can get your camera set up and we can get started right on time. So another question that I can give you on the exam, I can give you the sequence, and then I can say, tell me the amino acid, and then I can give you a mutated sequence, and I will ask you if the amino acid changed, and I'll give you the chart for it. So I think I gave you a similar question here. So something like this. I can give you original sequence, I can give you mutated, and then I can ask you which amino acid changed or if it didn't, or I might just have you write down the amino acid for both of these. For viruses, why aren't they considered alive? Why are viruses not considered alive?
Because they need a host to reproduce and all
I
that.
couldn't hear you, Nancy.
I'm sorry. Is it because they need a host
They need a
that
host. They
do
cannot replicate on their own. In order to do transcription, translation, they need a host and they use their machinery. They also don't grow, but the main thing is they cannot reproduce. I don't expect you guys to memorize any of these steps. Just make sure you know viruses are not considered alive. They cannot reproduce on their own. Then you have this lytic pathway and lysogenic pathway. I don't expect you guys to memorize the steps for these, but I do expect you guys to know the difference. So lytic pathway is the when the viruses break down the cell and they leave and they go impact other cells. Lysogenic is when the virus just hides inside the host DNA and replicates. So that's the difference between these two. And then you need antivirals for the viruses. Antibiotics not going to work for the viruses. Last thing I want to mention for this chapter is viriyoids. All I need you to know about viriyoids, they are circuits of RNA. They can impact the protein encoding. So basically, if this potato was supposed to express a certain protein, it doesn't do that because of there's interference from the circuits of RNA known as viriyoids. So that's chapter 7. Maybe you guys couldn't think of the questions before. Are there any questions now on chapter 7? Can you guys think of any questions in chapter 7? So Giselle, start and stop codon. So as long as you know that 3 letters stand for a codon, you should be able to count. So 3 letters stand for 1 codon. So I'm assuming no questions. Now I'm gonna go on chapter 8. And then I also wanna know how many people are still having hard time with the mitosis and meiosis. Maybe raise your hand if you're having hard time mitosis and meiosis. You guys are good? Okay, that's good. So can you guys tell me difference between mitosis and meiosis? Who can tell me difference?
Mitosis is when it's what happens when cells are dividing. Why meiosis is when sexual reproduction is being applied.
Yes, so mitosis is just cell division. It's cells dividing and dividing and dividing, and they divide for different reasons. They can divide in order for the development, or if you get a cut, you need new cells, skin cells there. They're gonna start dividing to repair. They can divide for regenerating lost body parts in amphibians. They can divide for asexual reproduction. I did a table for you guys. I want you to kind of go back. I compared mitosis, binary fission, and meiosis. And like George said, meiosis is referring to gametes and egg cells. So gametes are sperm and egg cells. If I were to ask you the location for the gametes, can you guys tell me for male and female? Where is the production of sperm cells happening and the egg cells are getting mature? For male and female.
The testes and ovaries.
Testes and ovaries. And also these gamete cells are known as germ cells. So you might see that term appear on the exam. Make sure you know what it means. And then there's something called apoptosis. Apoptosis. Can you guys tell me what apoptosis is?
Cell death.
It's pre-programmed cell death. I gave you an example of the chicken and the duck. Duck, they can swim. Chickens don't swim. They both have this webbing in the feet, but because of this apoptosis or pre-programmed cell death in chicken, there's no function for it. It just dies. These cells in between, they die because they don't swim. Whereas these remain because they swim. So the webbing remains here. So apoptosis is pre-programmed cell death. Then we have DNA replication. What are 3 enzymes that are important for DNA replication? Can you guys tell me the enzymes and the functions?
Helicase, DNA polymerase, and ligase.
Helicase, DNA polymerase, and ligase. Who can tell me the functions of those?
Polymerase is the builder. Ligase is the gluer. And I don't know if I get it. Gluer, yeah. It's glues. Its work is just to glue, to attach. Why? There are 3, right?
Yes.
I mentioned 2. The other one I can't remember the name, but I know the function. Helicase is the one that unzap, like it's detached. You
Yes.
know, detaches the sequences. Yeah, thank you.
So this is what I told you the day I did the lecture. I said DNA, double-stranded, it's like a book. You need to open the book in order to read the information. And that's done through helicase. Helicase opens it, it unzips it. And what the helicase is breaking is those hydrogen bonds, which is covalent bond. They're weak bonds. So it breaks those hydrogen bonds, it opens it. DNA polymerase goes in, it adds the complementary base pairs. When I say complementary, I mean if there's a G, it's going to add C. If there's a T, it's going to add A. Then ligase, if there are any breaks in between, there are any mistakes, it's going to go and fix the breaks. It's like a glue. Also, I have another term. What is semi-conservative? What does semi-conservative mean?
There's one original strand and one daughter strand.
So there's one original strand, and then you have one new strand or daughter strand, whatever you want to call it. That means it's semi-conservative. It has half of the information that's original, it has half of the information that was built later or new. What is binary fission?
It's another type of cell division that happens in prokaryotes and asexual reproduction.
So it's a cell division, binary fission and cell division that happens in prokaryotes, and the prokaryotes are bacteria and archaea. And it's asexual, which means it's going to give you exact same copy. You have this original parent DNA, and here you have 2 daughter cells. Both of them have exact same information. Now, if I were to say that if this cell gets maybe some new, some changes in the DNA. How is that possible? Even though it's binary fission, they divided and they had the exact same DNA. Now this got some changes. How is that possible? What are some ways it might be possible?
Mutations.
Mutations would be one. Can you guys give me another one?
It can take in genetic information from its environment.
It can take in genetic information, which is known as gene transfer. And if you were maybe manipulating this cell, you could do some DNA technology and manipulate the DNA. So that'd be third. Genetic engineering. Yes, Kawa. Okay, now I am on mitosis and meiosis. So I'm gonna do these together. I'm gonna try to maybe help you memorize it a little bit better if you're struggling with it. I know a few people, I think 2 people said they're having a little bit of hard time. So let me compare these. Actually, before I do that, I want to know if you guys understand cell cycle and cell division. If I ask you, give me the steps for cell cycle, would you be able to do that? Do you guys know what cell cycle means?
It's gonna go ahead, go ahead. Okay, it's gonna include interphase, mitosis, and cytokinesis.
Yes. So it's going to this cell cycle, or cell cycle means the whole cycle. It has interphase, then it has mitosis, and then it has cytokinesis. If I say, give me briefly the steps of cell cycle, you're gonna give me these 3. If I ask you to go in detail, then you're gonna say G1, S, G2, prophase, metaphase, anaphase, telophase, and cytokinesis. But if I say only give me the steps for cell division, then you're gonna say prophase, metaphase, anaphase, and telophase. So PMAT. That's for cell division. This whole thing is cell cycle. So make sure you understand the difference. Okay, then here I have mitosis. So first step for mitosis is prophase. It says early prophase, late prophase. And then you kind of want to read what's happening. And the reason there is early and late is because sometimes they look at it and this is happening, and sometimes they look at it, it's at this stage. So there is 2, but I just want you to write it as 1. So what's happening in prophase is this nuclear envelope is breaking up. So this nuclear envelope breaks up, the chromosomes condense. So you can see them when they're condensed, they're highly organized. And then the spindle fibers form and they go to the opposite ends. So that's mitosis. So let me try to do a comparison for that. So I'll do a table. So I'm gonna do a table, but I am going to say this is mitosis, and I'm gonna say meiosis 1 here. I'm doing this just so you guys can understand it. This does not mean that meiosis 1 and 2 are different processes. They're still the same, one single process, but I'm just doing this for comparison because you have prophase, metaphase, anaphase, and telophase in all of these. So I'm doing this just for the comparison. So first one is prophase. For prophase, for mitosis, we said chromosomes condense, nuclear envelope breaks up, and then the spindle fibers form. So that's what's happening in mitosis. So chromosomes condense, spindle fibers form, and move to the opposite end. And then also nuclear envelope breaks up. That's mitosis. Now let's look at meiosis. What's happening in meiosis? Here is meiosis 1 prophase. It says chromosomes condense, spindle forms, nuclear envelope breaks up. Exactly same thing. So you have exact same thing going on here for prophase. But in addition to that, there's one more thing that's happening, and that's crossing over. So that's something new. So you want to add that here, plus crossing over. So this is something That did not happen in mitosis because that's just cell division and this is sexual reproduction. So that's something new. Let's look at meiosis two. What's happening in the prophase? It says spindle forms, nuclear envelope breaks up, prophase two. So it's saying exact same thing again. Spindle forms and nuclear envelope breaks up. That's your meiosis two. So there's not much different between the phases, just few things. Then you have metaphase. Can anybody tell me what's happening in metaphase for mitosis?
Segregation. Sorry, they lined up in the middle.
So M for middle, maybe try to remember like that. The chromosomes, they line up in the middle and you have a single row. So chromosomes line in the middle. And it's a single row. That's metaphase for mitosis. Let's look like a metaphase for meiosis. So for meiosis one, here is your metaphase. It says paired homologous chromosomes line up in the middle. Still same thing going on, but they're paired and they're homologous. Hopefully you guys know what homologous means. And you have double row. So I'm gonna say paired homologous chromosomes align in the middle and you have double row. So it's not single, it's double. And it's paired homologous chromosomes.
Same chromosomes. That's a
Yeah.
homologous same.
Homologous means they have the same length, they have the genes at the same spot, and they look the same. So that's homologous. You have a double row. Let's look at meiosis two. For metaphase, chromosomes align in the middle, and you again have a single row. So that's exactly same as this, as mitosis. So I'm gonna just copy paste that. And you have single row again here. And then you have anaphase. So let's look at anaphase. For mitosis, it says that they start splitting. Anaphase, maybe think of A as a wave. They start moving away from each other. So here it says that sister chromatids separate and move to opposite poles of the end. So anaphase, sister chromatids separate and move to opposite ends. Then you look at the anaphase in meiosis 1 and 2. Here is your anaphase. They're still separating and they're moving away, but the chromosomes, they're still looking like X's, whereas in mitosis, they were looking like V's. So they look like letter V in mitosis. I'm gonna say look like letter V. Whereas here, they still look like X's for meiosis 1. For meiosis 1, they're still looking like X's here. So they're homologous chromosomes. They separate to opposite poles. Sister chromatids, they remain joined. They don't break yet. You still have the X. So actually, I'm just gonna type it. So, so I'm gonna say homologous chromosomes move to the opposite ends. And they look like letter X. Sister chromatids remain joined. That's something new. This is different. Then you look at meiosis 2, anaphase. Now you have sister chromatids separating. Now they start looking like V's. So this is exactly same as this. They start looking like V's for meiosis 2. Then you have telophase. For mitosis, you have telophase, nuclear envelope reappears and spindle disappears and chromosomes decondense. That's what's happening in the telophase. So nuclear envelope reforms or forms chromosomes decondense, and one last thing is spindle disappears. So you don't need the spindles anymore. Spindle disappears. That's telophase for mitosis. Then you have telophase for meiosis. One, nuclear envelope reappears, forms again around the chromosomes, and the chromosomes decondense and spindle disappears. So you have exact same thing happening here in meiosis 1. And then in meiosis 2, you have nuclear envelope reappearing, but now it's happening around the daughter nuclei. Chromosomes still decondense, spindle still disappears, but the only difference is that it's happening around daughter nuclei because, again, meiosis 2 gives you haploid cells. So I'm going to say around daughter nuclei. So that's, again, different. And then you have cytokinesis. Can you guys tell me how many cells you guys get after cytokinesis in each? What is the number of cells you get for mitosis, and then at the end of meiosis 1, and then for meiosis 2?
You get 2 for mitosis, 2 for mitosis 1, and 4 for meiosis 2.
You have haploid. Does this help a little bit, Baron? I know you were having a little bit hard time with this. Any questions on this?
Seeing everything really helps.
Okay, okay. So if you guys need to take a photo of this, go ahead and do that. So
I think this is the best.
What was that, George?
I think this is the best way to kind of, state it in order to have everyone knows about it.
Yeah, so there's not much different if you compare all of them, but because it's a lot, sometimes you might get overwhelmed with the information, but it's the same information if you really compare it. But I really, really want to make sure you guys understand this is one process. It's not different processes. You have meiosis 1 and meiosis 2 happening in a single process. But I did this just for the comparison so you don't, you can memorize it a little bit better. On the exam, you can expect these photos. I can give you a photo and I can say, tell me which phase is happening. If it's meiosis phase that I'm showing, I'll say, tell me what phase is happening in meiosis 1 or meiosis 2 or mitosis. So you can expect the photos. Again, the photo questions in the exam are worth a little bit more. Same thing for the chart, for the DNA information chart, the amino acid chart. It's, I think, worth at least 7 to 8 points on the exam. So make sure you guys can really do those charts. And then there's crossing over. Crossing over is when there's mixture. You get a mixture from crossing over. And you have these two homologous. Homologous means they are the same length, they look the same, they have the genes at the same spot. The genes might have different origins, which means alleles, but they're still, the same gene. And then if they're close to each other, there's crossing over that can happen. And this crossing over happens during prophase 1. This is important. The crossing over happens during prophase 1. And then there is an independent assortment. Can somebody tell me what independent assortment means?
The way the sister chromatid will be arranged.
The way sister chromatids will be arranged. So if you guys remember, I said if somebody have a child and they have a child the second time, there's 1 in 8 million chance the sperm with the same genetic information is going to fertilize the egg. So the random independent assortment gives you these random sister chromatids. They are arranged randomly. Maybe this one has 1 red, 2 blue, this one has all 3 red, but all this is happening at random. So that's the independent assortment. It's happening randomly. Now, if I were to say, tell me the difference between monozygotic and then dizygotic twins. Could you do that?
I believe monozygotic twins will be The ones that are identical.
They're identical.
Why?
Why are they identical?
Yeah.
What is the process that's happening in monozygotic versus dizygotic?
Monozygotic
Just
twins, it's one egg that gets split into two, and then with dizygotic twins or the fraternal, it's two eggs that happen to be fertilized at the same time.
Yes, so monozygotic, they look the same. That's because there was only one egg and one sperm, and this egg got split, so it had the same genetic information. Dizygotic, you have two eggs, you have two sperms, and after the sperm fertilized this egg, you have first zygote here, and then the same thing for the second zygote. Now you have two embryos, but they don't look the same because it's a different sperm that's fertilizing this egg versus this egg, so they don't look the same. So that's dizygotic. Then there is something that's called non-disjunction. We just went over meiosis. Sometimes when it's going through meiosis, it doesn't happen properly, which means the chromosomes don't divide equally. This picture is showing meiosis 1. Something happened in meiosis 1. What is the phase where the chromosomes separate in meiosis 1? What is the name of that phase? Where the chromosomes separate or they move away from each other?
Is that the
telophase.
Anaphase. So if it's meiosis 1, it's going to be anaphase 1. During anaphase 1, these chromosomes, they fail to separate. They couldn't separate equally. This one doesn't have anything. This one has double. That's non-disjunction. What is the result? These are your gametes. This is abnormal one. It's supposed to have only one. Now you have two. Abnormal one again. These are just empty. So all four are abnormal. If during meiosis 1, the chromosomes fail to separate evenly. And then you have extra pair here. Now this will be the zygote after it fertilizes the egg, and there's going to be issue. There's going to be a genetic disorder. If it happens fine, if they separate fine during meiosis 1, but then during meiosis 2, they fail to separate for this one, then this is your gametes. You have two normal ones, and then you have two abnormal ones. Now you could go ahead and do the Punnett score and say, oh, there's 50% chance this child is going to have a genetic disorder, or he's not going to have a genetic disorder, because there's a 50% chance this sperm can fertilize the egg. 25 for this, 25 for this, and then there's also 50% chance of receiving abnormal sperm with the abnormal number of chromosomes. So that's called non-disjunction, and it's an error that happens in meiosis. It can happen during meiosis 1, it can happen during meiosis 2. I don't expect you guys to memorize this table, so don't worry about that. And then if you have number 21 extra chromosome, then that's Down syndrome. So that's the one we talked about. Any questions on chapter 8? Oh, I finished chapter 9. I think I started from meiosis and I went down. I need to go back there. So let me kind of see what I missed on chapter 8. The only slide I want to go on for chapter 8 is the tumor. Can you guys tell me the difference between benign and malignant tumor?
benign is a not active tumor and malignant is an active one?
So benign is so tumor. If you were to define tumor, it's uncontrolled cell division. Their cells are just dividing and dividing. They divide out of control. Then you have two types, benign and malignant. Here, this example is saying that there are some cells that are dividing uncontrollably, but they're remaining in this place, in your connective tissue. If you don't know what that is, just think that maybe it's in your lung. So I'm just going to give an example. This is happening in your lung, but this is only happening in your lung. It's not going anywhere in your body. You have your blood vessels going to the lungs. Then some of these cells break off and they get into those blood vessels. Now those blood vessels are going everywhere in your body and they go to your kidney. Some cells go in your kidney, start causing a new tumor there. Maybe they go to your liver. Some cells go to your liver and start causing a new tumor. So malignant, it can go to other parts of your body and start a new tumor. This one stays in one place, but it doesn't mean it's not dangerous. It can be dangerous as well. So this stays in one place and this goes into other parts of your body. So tumors throughout the body, malignant, And then if it's not spreading, it's benign. That was the only thing I missed on chapter 8. I need to go back to chapter 9. I missed the beginning of chapter 9. Can you guys tell me some benefits of sexual reproduction over asexual reproduction?
Genetic diversity.
Genetic diversity. When is that genetic diversity useful?
If there's like a virus or something like in the potatoes or something where since they were all genetically identical, one virus was able to wipe them all out. And it also helps with evolution where maybe since there's cats here, like darker colored cats would survive longer in certain environments than lighter colored cats.
Yeah, so that genetic diversity is important when the environment is changing. If there's something, new disease pops up, and maybe if you have genetic diversity, it might be able to get through that. So that's some benefits of sexual reproduction versus asexual. Then I gave you guys some vocabulary words. George already defined homologous. Homologous means they look alike, they have the genes at the same spot, and they are same length. That's homologous. These two chromosomes are not homologous. What do you call chromosomes from 1 through 22? 23rd is the sex chromosome.
Autosome.
What is the sex chromosome for females?
Sex chromosome.
So what is the sex chromosomes? What is the letters for females?
X and
For
X.
female is XX. For male
For
is
ma-
XY.
Yes, for males it's X and Y. If you look at this X and Y, you can see Y is shorter. Y is shorter than the X, and it's lighter in weight, and that's why it says and there's more males born versus females, because the sperm with the Y chromosome is able to swim faster. They're not homologous, they're not the same length. And then there was another word I gave you, was karyotype. What is karyotype?
The way that nucleus been organized.
So karyo means nucleus and type means to organize. If you open the nucleus, you see all 23 pairs, and they are maybe organized according to the length. That's karyotype. This is showing you homologous pairs look alike. Can you guys tell me definition for allele?
A particular characteristic from two parent. Okay, from one parent. Like different versions of genes.
Yes. So different version of a gene. Here you have you have one chromosome here, another homologous here. And I said during class, this is for cake and this is for ice cream. Big A gives you chocolate cake. You have chocolate cake here, you have chocolate cake here. Big B gives you vanilla ice cream. And little b gives you chocolate ice cream maybe. So this gene is still for ice cream, but it's a different version of ice cream. So alleles are different versions of the gene. They're not different genes, they're just different versions of a same gene. Can you guys define a zygote?
Zygote is when a sperm and egg has been fertilized.
So when the sperm fertilizes the egg, gives you a zygote. This has haploid cells. This has haploid cells. Once the fertilization happens, it's diploid. So zygote is first new cells. Then zygote goes through the mitosis, starts dividing and gives you, development and growth. And we already covered this part, so I'm not gonna go back here. So let me